Hello guys, welcome to my channel SwathiPharmaJnan. Today's topic is macrolides. Macrolides are also under antibiotics. Macrolides are a group of antibiotics which contains macrocyclic lactone ring. Lactone ring is usually it contains 14 to 16 carbon atoms to which deoxy sugars are also attached. These macrolides are obtained from Streptomyces species. Examples of macrolides under antibiotics are erythromycin, azithromycin, clarithromycin. The structure of erythromycin contains lactone ring. Two deoxy sugars are attached here. Various substitutions like hydroxy group, ethyl group, methyl group, and dimethyl amino group, methoxy groups are attached. Uses are used against gram-positive and gram-negative bacterial infections, to treat syphilis and diphtheria, to treat pertussis, which is whooping cough that can cause severe coughing. The next drug under macrolides is azithromycin, which is one of the important drug used for various bacterial infections. The structure of azithromycin includes the lactone ring, and two deoxy sugars are attached to the lactone ring with various substitutions. Uses of azithromycin is azithromycin is active against many respiratory tract pathogens including pneumococci, mycoplasma and chlamydia species. And this azithromycin is used to treat throat infections and non-gonococcal urethritis. It is expensive drug. Azithromycin is one of the expensive drug. It is used to prevent bacterial complications in AIDS also. Next one is clarithromycin. Clarithromycin is another macrolide antibiotic. The difference between clarithromycin, azithromycin and erythromycin are various substituents. So if you will practice the lactone ring with the substituents and deoxy sugars, then it will be easy for you to understand and draw the structure during exams. Uses of clarithromycin is to treat bacterial infections which are affecting skin and respiratory tract. Clarithromycin is combined with other drugs which are used to treat stomach ulcers caused by H. pylori. The mechanism of action of macrolides is macrolides inhibits protein synthesis by binding to the 50S ribosomal subunit. Suppression of these macrolides will inhibit the protein synthesis. How they will inhibit the protein synthesis? The drug will go and bind with the 50S ribosomal subunit which is involved in the protein synthesis. Suppression of RNA dependent protein synthesis by inhibition of translocation of mRNA. The process of translocation of mRNA will be inhibited by suppressing RNA dependent protein synthesis. Macrolides are bactericidal which will kill the bactericides at high concentration against very susceptible microorganisms. If very susceptible microorganisms are present at high concentration, they will act as bactericidal antibiotics. The diagrammatic representation of mechanism of action is 30S ribosome and 50S ribosome. The drug macrolide will come and bind with the 50S ribosomal subunit, and this 50S ribosomal subunit which is involved in the protein synthesis, mRNA template is involved. This 50S ribosomal ribosomal subunit is important in the protein synthesis, so that will inhibit the translocation of mRNA. Finally, protein synthesis will not occur. The various side effects of using macrolides antibiotics are nausea, vomiting, abdominal pain, chest pain, dyspepsia that is burning sensation, flatulence, vaginitis, inflammation of vagina, nephritis, inflammation of nephron, dizziness, headache, photosensitivity, rashes and pruritus. This completes macrolides topic. Thank you.